Hello Bruins, this is Mrs. Farouk, and today we're going to talk about meiosis. Now before we also talk about meiosis, there are key terms that we need to know. For example, what is the difference between chromosome and chromatin? What is a homologous chromosome versus a non-homologous chromosome? And the difference between phenotype and genotype, and diploid versus haploid. So at the end of this presentation, you should be able to describe the process of meiosis and explain how variation is maintained by meiosis. Now, if you look at these two couples and you look at the children, can you identify what two kids belong to which couple? And see if you guessed it right. So you could see there that these kids share similarities to their parents, and yet each sibling is different, and this is all because of how your chromosomes are mixed up during meiosis. Now, again, a quick review between asexual reproduction and sexual reproduction. So we spoke about asexual reproduction when we spoke about mitosis, and if you can remember, what are the advantages and disadvantages? So key thing about asexual reproduction, again, advantages: you don't need a mate, don't spend energy courting a mate or finding a mate. Usually occurs faster, the rate of reproduction is faster, and usually there's not much energy expended for offspring care. Disadvantage, less variation, and variation is really important, and this is important to evolution. And when we talk about evolution, we mean in terms of to survive, to reproduce. Okay, so that's important. In sexual reproduction, advantages is key is variation. Okay, the more varied you are, the greater survival rate. Disadvantage, energy is required to find a mate. Usually there's more offspring care, parental care is required. All right, and it is a slower rate of reproduction compared to asexual. Okay, so again, when we talk about sexual reproduction, we're going to talk about how those two sources recombine to help get that variation. All right, so life cycles of animals consist of both haploid and diploid stages. Now, what does that mean? If you remember, the word haploid means one set of chromosome, and diploid means two sets of chromosomes. So notice 2n represents two sets and the number of chromosomes, and haploid means half that chromosomes. So meiosis is the process to produce gametes by reducing the number of chromosomes in half. Okay, and where does it happen? It happens in the sex organs, ovary for the females and testes in the males. Okay, and what is the product? And the products is the gametes, or we can also use the word sex cells, or in particular egg and sperm. Okay, and what is fertilization? So fertilization is when the egg and sperm unite, and the result of fertilization is a zygote, which we called a fertilized egg. Okay, so you have a combination of two sources of chromosomes, one from the egg and one from the sperm. Then again, reminder, haploid is one set of chromosome and diploid is two sets. So in diploid, one you get from your mother and other set you get from the father. that's contributed from the sperm, and the mother is the egg. So if you can see, this is a life cycle. So notice most of the life cycle in animals is the diploid stage, and only part of our haploid stage is gamete formation. So reviewing, if you remember, chromosome is how DNA is packaged, and we see mitosis, and here we have DNA, and DNA is packaged into nucleosomes, and that nucleosome is further looped into chromatin, and then chromatin is further condensed into chromosome. So if you can remember, we see chromosome during cell division. Now here's a new term here, is homologous chromosome. What's the difference between homologous and non-homologous? So homologous chromosomes is that you have a pair of chromosome, one you get from your father and one you get from your mother. Now these are not identical. So here, for example, is chromosome pair number 2. They're not identical because they don't have the exact identical genes, but they have the similar genes on both chromosomes, but they might have different versions. So that's what homologous means. Now comparing, say, chromosome 2 to another chromosome, say chromosome 9, we say they are different chromosomes. Okay. Now, have you ever seen a karyotype? So karyotype is basically a picture of your chromosome. So they basically take it, arrange it in the chromosome. So chromosome pairs number 1 to 22 is called a autosome. And then the chromosome that determines your sex is called the sex chromosome, and that's the X chromosome and the Y chromosome. So here in this karyotype, so there's your autosomes 1 to 22, and there is your sex chromosome. So this individual has X and a Y, so it's going to be a male. Now can you tell me the sex of this individual? Notice it has 2 X's, and yes, this is a female. Now sometimes what happens, there can be a mistake during meiosis, and when a mistake usually happens, say during anaphase, where the chromosome does not separate properly, and that causes an abnormal number of chromosomes, and that's called aneuploidy. In aneuploidy, you can have chromosomes where you are missing a chromosome, where here you see n-1, usually a syndrome like monosomy results, or you can have an extra chromosome called trisomy. In animals, this can be lethal or cause like a syndrome. So here, for example, look at the karyotype. Do you see anything wrong? Notice chromosome 21. Again, this is a result of a non-disjunction at this chromosome, and you have an extra chromosome, and that's called trisomy. How about here? Notice you have an XXY. So this person is going to be a male, but with the extra X chromosome, and this is called Klinefelter Syndrome. Turner syndrome, as you can notice, only has 1 sex chromosome, 1 X chromosome. Jacob syndrome, here you have an extra Y chromosome. Now sometimes if you have in other chromosomes, usually the embryo is not viable, does not live, but then in certain chromosome, like chromosome number 21 or the sex chromosome, you'll have, or even chromosome, I think number 13, you'll have some kind of a syndrome. Now, another few words that you need to know is what's the difference between somatic cell and germ cells. Somatic cells are, another word is a body cell. Okay, body cell that's not a sex cell. So for example, kidney, brain, kidney cells are made of nephrons, or brain cells are made of nerve cells. Muscle is made out of muscle cells. Now, germ cells give rise to gametes, or in other words, sex cells. Okay, and these sex cells are in female egg and male's sperm, and when it unites, you form a zygote. Okay. Now, looking at the cell cycle, if you remember in mitosis, we learned the cell cycle includes interphase, And during S phase, you have DNA replication. And then when it's ready to divide, the nucleus divides, and that's mitosis. And when the cytoplasm divides, that's cytokinesis, producing two identical cells. Okay, identical to the parents. Now, the cell cycle of a mitotic cell, again, this is happening in a germ cell in ovary or testes to form a gamete. So it too will go through G1 S phase where DNA is copied. And remember, one chromosome is like from the mother, and one chromosome you have from the father. So when this replicates, you'll have homologous pair. And then it'll go through a nuclear division, meiosis 1 and meiosis 2, and this time producing 4 haploid cells that are genetically unique. Right? And you started off with a diploid germ cell. So in humans, we have 46 chromosomes. Our diploid number is 46, and by meiosis, you'll have 23 in each gamete. Okay, so basically, meiosis cuts the number in chromosome in half. So here, this kind of summarizes what happens during interphase. So you have a chromosome, say, from the mother and one from the father, and each pair of chromosomes then will replicate. And now you each has sister chromatids. So this blue is a chromosome with identical chromatids, and this red chromosome has 2 identical chromatids. Together, this is called a homologous pair. Okay, and this is something you don't see in mitosis. And in meiosis 1, after meiosis 1, you cut the chromosome in half, right? And then you further cut in half again, and you produce haploid cells. And if you can see, between meiosis 1 and 2, there is no replication. Okay, so basically, during meiosis, you are replicating the DNA, you're pairing up those homologous pair, and then separating and divide, and then separate. So there's 2 phases, 2 sets of division. All right, so let's go through the phases. So this is what you have in your notes. So the first phase, of course, is interphase. And key things that happen in the interphase that you have to remember is DNA replication. And remember, here you will have homologous pairs after replication. And you know it's interphase, you see the nucleolus, you see the nuclear membrane, and remember your chromosome is in the form of chromatin, right? And prophase, now here you gotta use numbers. Prophase 1, a lot of things happen in prophase 1. In prophase 1, here our chromatin is becoming chromosome, getting more condensed, right? And another big thing is something called synapsis, and synapsis means pairing up. So here the paternal chromosome is going to find its pair and pair up. So say this is chromosome pair 1. Okay, so what is pairing up? Homologous chromosomes are pairing up. So we call this homologous pair. Another key important thing that happens that's different from mitosis is crossing over. And crossing over is basically the exchange of segments of chromosomes. So you see this blue chromosome is exchanging with this red chromosome, and so not between the non-sister chromatids. So you will have one that has exchanged and one you will have parental chromosome. Now, then in metaphase 1, what lines up? Homologous pairs line up. and then anaphase 1. Now you have homologous pair or chromosomes being pulled apart. So chromosome or homologous pair separating. Okay. Now other words you might also hear to describe synapses or homologous pair is you have 4 chromatids. So 4 chromatids is a tetrad. And notice here 2 chromatids is called a dyad. Dyad means 2. Okay, so let's see if we got all that information. So here, interphase, chromosomes duplicate. Prophase 1, homologous chromosomes pair up, called synapses. And you have exchange of chromosomes segment called crossing over. Okay, very important too, variation. We're going to go over this later on. And then metaphase 1, homologous pair line up, or tetrads line up. Homologous pairs separate, where each chromosome is going to opposite poles. Then you don't have anything between, no replications happening or anything, but then it goes straight to meiosis 2. And notice now we're looking at 2 cells. So here we have telophase 1, right? And there's your cleavage furrow, and now that is splitting, right? So now you have 2 cells here, and then this is prophase 2. And in prophase 2, there's no synapses or crossing over. It's just like mitosis. So very similar to all these will be very similar to mitosis. Okay, so you see the nuclear membrane disappearing, your spindle fiber forming, and here, just like in mitosis, you have chromosomes line up. And here, each chromatid separates, moves to opposite ends, and then here you have telophase, and again now our chromosome becomes chromatin, and it's a haploid cell. It has half the number of chromosomes from a germ cell. So here again, metaphase 2, and you know that you see a dyad, you don't see a tetrad, you see chromosomes lining up. And this is anaphase 2 because chromosome is being pulled apart, and each sister chromatid is going to the opposite end. And then you have telophase 2. So basically meiosis 2 is like mitosis, except you are generating 4 unique cells. So let's see if we got everything. Okay. So how does meiosis generate genetic variation? If you remember, you have a chromosome, one pair from your father and one from the mother, right? And key phase is prophase. So during prophase, you're having crossing over, exchanging segments of the chromosome, and then it is being separated or segregating, going to different cells, and that further gets separated. So here, if you can see, you have 4 genetically unique cells. So take some time and practice on describing what happens in crossing over during prophase. So here you have homologous chromosomes. They pair up, which is called synapses. Okay, and when they pair up, then they're ready to cross over between the non-sister chromatids. And when I say non-sister chromatids, I mean here's the non-sister chromatids. These 2 green and these 2 orange are sister chromatids. Okay, the describe what happens during crossing over using these terms. If you want to pause and practice using these terms. So here you have chromosomes. The different genes or alleles are represented by letters. So here's a homologous pair. And between the non-sister chromatids, segments of chromosomes are exchanged, producing 4 genetically unique chromatids. And that contributes to genetic variation. So if you see also in metaphase how the chromosomes line up. So metaphase can be a key phase in terms of generating variation. So in metaphase, how those chromosomes line up will determine what chromosome will go to what cell, and that will determine what cell at the end will get what type of chromosome. So how does meiosis differ in human males and females? So gamete formation in males is called spermatogenesis. So here you form 4 sperms that are genetic haploid, meaning they have half the number of the germ cell. So the germ cell will start off at 46 chromosomes, and these haploid each will contain 23 chromosomes, and each genetically unique. In females, it's called oogenesis. Yes, it is with 2 O's. And you start off with a germ cell, an egg germ cell, with again 46 chromosomes. Now, at the end of meiosis 1, you're going to have an egg cell, and it produces a non-functional cell called a polar body, which will basically disintegrate. And then, and this is the key difference, and after fertilization, it completes meiosis. So after fertilization, it'll go through meiosis 2, and again producing another polar body. And notice most of the cytoplasm goes to the egg. So cytokinesis is not proportional and does not divide the cytoplasm evenly. Okay, so let's compare mitosis and meiosis. Again, think of the purpose and mechanism. Mitosis, key difference are in prophase. Notice you see a dyad, 2 sister chromatids per chromosome. And meiosis, you see homologous pair, you see tetrad. Metaphase, chromosomes line up. In metaphase and meiosis, homologous pair line up. Okay, so chromosome number is key. In mitosis, it's a clone, same number of chromosomes, and in meiosis, you're cutting the chromosome number in half. Okay, so let's quickly review our life cycles. So here's a life cycle of human, right? So gametes are formed by meiosis, producing haploid cells. And the only time where haploid is in a sperm or an egg production, and when those two unite is called fertilization. In fertilization, then you produce a diploid cell, which is called a zygote, and that through mitosis develops a embryo, becomes a fetus to a baby. So most of the animal cells, diploid is the major part of its life cycle. Okay, so if you're given a life cycle here, gametes that are produced from meiosis. So here's meiosis, and when fertilized, or fertilization, produces a diploid zygote, and most of its life is its multicellular life is in the diploid stage. Okay, the majority of its life from a diploid organism. So this is an example that we find in a lot of the animals. Now here, notice the haploid stage is more dominant. So here gametes are produced again by meiosis. Okay, and after fertilization, very briefly, you'll have a brief life as a diploid. from a unicellular diploid organism. And this kind of life cycle you usually see in a fungus. A fungus usually spends most of its life cycle as a haploid. Okay. Now another type of life cycle is it alternates between diploid and haploid, and this you'll see in plants. So here again, by meiosis, it produces these haploid cells, and so it'll alternate between its haploid life, and when it fertilizes, when these gametes fertilize, it produces a diploid. And then part of its life cycle, majority of the time, it could be in haploid. I mean, this is a diploid or in haploid, so it alternates. Okay. So review and come with questions, and then in your notes do the chart with the differences between mitosis and meiosis. Study the PowerPoint and make a chart. So here, for example, you have a summary of the differences between mitosis and meiosis. Key difference note, you have prophase, what happens in mitosis versus meiosis. That's a big difference where there is no crossing over or synapses in mitosis. Metaphase, what is lining up? Here you have chromosomes that are dyad line up. In metaphase 1, you have homologous chromosomes pairing up and lining up in metaphase 1. And what do you produce at the end? If you're starting with 2N in mitosis, you end up with 2 cells that are clones. In meiosis, you end up with half the number of chromosomes that are genetically unique. Thank you for listening, and come to class with questions.